Hello and welcome to Naad Education. This series of lecture is to clear our basics of biology. And the topic we address today in lecture 5 is Archaebacteria and the three domain classification. So first we will talk about what is Archaebacteria and then we will dive into the three domain classification. So what is Archaebacteria? Archaebacteria were first classified separately from bacteria or in prokaryotes. There was a classification that distinguished Archaebacteria from Eubacteria in 1977 by Carl Weiss and George E. Fox based on the following features. So the eubacteria and the archaebacteria differed in the sequence of 16 srRNA genes, which is the most important point in this. And then further studies revealed that the archaebacteria did not have peptidoglycan in their cell wall, and there were some other different coenzymes present in the archaebacteria, especially with respect to translation and transcription of the DNA in them. So these were the properties based on which the archaebacteria were separated from the eubacteria, and here is a diagram in which you can see its structure and some points. We move ahead and talk about further some general properties of archaebacteria. So archaebacteria and eubacteria are generally similar in size and shape, although a few archaebacteria are of very different shapes, such as a flat square cell of genus Haloquadratum. So flat square cells are not seen much in eubacteria, but in archaebacteria this genus does also have this shape, which is slightly different from the eubacteria that we see. The genes and enzymes and the metabolic pathways of archaebacteria greatly resemble that of eukaryotes, especially in transcription and translation. That's why we mentioned the point of the coenzymes before. And then archaebacteria have other lipids such as archaeoles present in their cell membranes. So these, not other lipids, ether lipids. So these ether lipids such as archaeoles, which are unique to archaebacteria, make their cell membranes little unique. After that, archaea can use sugars, ammonia, metal ions, and even hydrogen gas as energy sources. Archaebacteria, based on where they were first found, they were found in places of extreme temperatures and conditions where generally bacteria won't survive. So even in oxygen deprived conditions, archaebacteria can use other sources such as these mentioned here, for example, ammonia, metal ions, hydrogen gas as energy sources. Archaebacteria do not form endospores like bacteria for reproduction and reproduce by binary fission, fragmentation, and budding. So archaebacteria have not been seen to use endospores for reproduction, but eubacteria do show that mode of production of endospores for reproduction. And archaebacteria as pathogens have not been studied much and not been discovered much as well. But does not mean that archaebacteria are only found in trenches or in very extreme conditions. They are widely spread out in the world. Let's come to now, once you have understood what are the properties of archaebacteria and how are they found and what are the differences from the bacteria, from eubacteria, we now come to the three domain classification. The three domain classification was proposed by Carl Weiss, Otto Kandler, and Mark Wheelis in 1990. The key difference from other classifications is that the archaea, previously called archaea, is the same thing here, but I think I should have mentioned here that archaea right now is a domain. Previously it was called Archaebacteria, not archaea. So they split up from bacteria as completely different organisms. They were not just categories of Monera. So Carl Weiss could identify properties that allowed him to separate Archaebacteria from Eubacteria completely in two different domains or two different super kingdoms. So we're not just part of bacteria and two types of bacteria anymore. Archaea were first classified separately from bacteria in 1977 by Carl Weiss and George E. Fox that we have mentioned before, based on differences in 16S rRNA gene sequences and other evidences. We have talked about this in the previous slide. The three domain classification adds a level of classification above the kingdom. So earlier you must have seen in the previous lecture videos that we were talking about 5 kingdom classification, 7 kingdom classification, 6 kingdom, 2 kingdom, 4 kingdom. But here there is additionally just not the kingdoms. Kingdoms come under these three domains, and domains are a level above the kingdoms. So these are somewhere close to what you can call super kingdoms, or under which various kingdoms come. So three domains of life. This is how Carl Weiss designed his classification. Here are some properties in which the archaebacteria, the bacteria, and the eukaryotes differ from each other. The three domains differ from each other in these properties. So this is taken from Wikipedia. So when we talk about cell membrane, ether-linked lipids are very common for archaebacteria. They are not present in bacteria and eukaryotes. Cell wall is somewhere similar between archaebacteria and bacteria, and there's sometimes something called pseudopeptidoglycan that is present in archaebacteria that is not there in bacteria, that forms a unique cell wall for archaebacteria. And then peptidoglycan is also missing in the archaebacteria. There is something here, we can see it is mentioned that pseudopeptidoglycan is present. Gene structure, they are somewhere similar to the eukaryotes compared to the bacteria, but the size wise they are similar to the bacteria. Internal cell structure, no membrane bound organelles both in the archaebacteria and eubacteria, and the eukaryotes there are membrane bound organelles. We will make a separate video for differences between prokaryotes and eukaryotes that has been asked. Metabolism wise, you can see methanogenesis is one of the way. Mainly archaebacteria rely on anaerobic respiration, and methanogenesis is one of the modes for anaerobic respiration. So it will use methane or other elements such as hydrogen gas and other things for creating energy. Bacteria can use aerobic, anaerobic respiration, fermentation, and so many other ways. And eukaryotes can use to do photosynthesis, again similar to bacteria, but yeah, similar to bacteria. Reproduction, one point that we just talked about before was the production of endospores. So endospores are not there in archaebacteria. That is not mentioned on this slide, but you can mention it, that archaebacteria do not use endospores for reproductive purposes, but bacteria do, and eukaryotes use sexual and asexual reproduction. Horizontal gene transfer is again characteristic of these two bacteria, the archaebacteria and eubacteria. Protein synthesis initiation, the methionine is common for archaebacteria and eukaryotes, and formyl methionine is specific to bacteria. RNA polymerase, there are multiple in eukaryotes but only one and one in archaea and eubacteria. And the last point is not, it's a little technical, but we can skip this for now. Let's also talk about the two domain classification. It's a very interesting thing to note that the three domain classification did rule for some time, but there were very interesting studies later on, and that is where the two domain classification has come up. So there's a way of classifying organisms by holding only two domains. So all the organisms in the tree of life are classified into two domains, bacteria and archaea. The two-domain classification was preceded by the eocyte hypothesis by James Lake in the 1980s. So what was this eocyte hypothesis? The eocyte hypothesis in evolutionary biology proposes that the eukaryotes originated from the group of prokaryotes called eocytes based on the rRNA sequences. These were based on the studies of rRNA sequences, based on which they concluded that the prokaryotes, um, the eukaryotes emerged from the prokaryotes, and more specifically, the eukaryotes emerged from a group of archaebacteria. So, not every the type of bacteria that gave rise to eukaryotes were the archaebacteria type of bacteria, and those were initially called the eocytes. For later on, these eocytes were classified into a different group altogether. The main thing, if you look at the fourth point, the two domain system became more widely accepted after the discovery of a large group of archaebacteria called the Asgard in 2017. These Asgard, this group of archaebacteria resembled a lot with the eukaryotic cells that we see today, and they are supposedly the ancestral population, ancestral type from which, ancestral bacteria from which the archaebacteria, from which the eukaryotic cells first came about. The Asgard was suggested as the evolutionary root of the eukaryotes, as they contain the genes that encode the proteins that resemble eukaryotic signature proteins. So based on these studies, recently the two domain classification has taken um, a hold, in which the entire tree of life is classified only under bacteria and archaea, the two domains, and the eukaryotes come under the domain archaea. So with this we come to an end to this lecture. I hope you understood what all topics there are, and I hope these pointers are helpful for you in your notes. And if you have any confusion or request for notes or any other help with this topic, then please put your requests in the comment section. I will happily make a video on that as well. And please share this video with your friends. Like the video if you like it. Subscribe to the channel for other videos, and check out this series for whatever basic simple notes that you might require for studying biology. Thank you so much. Have a nice day. See you soon.